So, hi everyone. Good morning or afternoon, depending on your time zone. First, let me introduce myself. I'm Lucie Saint-Joanis, data manager at Bioaster, and today I want to talk to you about the data management at Bioaster we do principally with the LabKey Server SDMS. So I will start with some context and overview on what is Bioaster, what we do, then the challenges that we have into data management here, how we choose LabKey as a solution, and then some conclusion and perspective. So first, about the context and overview. Bioaster is a technological research institute which is dedicated on 4 specific fields: vaccines, antimicrobials, microbiota, and diagnostic. So it's kind of 4 of the trending pillars, I would say, into healthcare. A little bit about history. Bioaster is a non-profit organization that was created in 2012. And we were created by public institute and private institute. So you can see that for the public part, there were some French organizations that were involved, like CNRS and INSERM, or some universities. And for the private part, you can see that our founders are Danone, Boehringer Ingelheim, bioMérieux, Institut Mérieux, and Sanofi. We are also we are supported by the government, the French government, and so we grew since the last 10 years. We are almost 100 employees, based in 2 locations, so Paris, capital of France, and in Lyon. And through the 10 last years, we have worked with more than 100 partners, academic or not, and we principally work on collaborative projects with them. So most of the time, how does a project work at Bioaster? Some of our partners come with a biological question, and we have, I would say, 6 different skills, fields, and teams that you can see here that can go from the clinical operation, where our team is handling the sample and clinical management, to the omics hub, working on the different omics you can see there. And of course, the data management where I'm in. And using all of those skills, we try to find a biological answer to the question we were asking. So now, going a little bit into details, I will work about the challenges we encountered. When data management started at Bioaster, we had kind of a wish list, what we wanted to do with the data. So we wanted a way to centralize the data and metadata into one place, to have a secure access of this data, be sure that they are safe, a way to share all of these results with our different partners without having some issues about how can we share with this partner and not with this one. And we wanted to have a platform internally so we could manage everything. And we also wanted a way to automatize the data workflow, but still be flexible, because we will see that we have some really different projects, and that we need to have a way to do things one way or another. And last but not least, a very important part for me as a data manager is to apply the FAIR principles to our data. So make sure that our data are findable, accessible, interoperable, and reusable. And that is the big part. So now I will show you some examples on how we are handling data and how we need to adapt between one project to another. So on this example, we had a partnership with an hospital. This hospital collected blood samples from us for us, sorry, and of course the metadata linked to them and the clinical data. And we had the partner who received these samples and the data associated with it. This partner needed to do the quality check and the sample preparation to be sure that everything was fine and that we could go on with our process. They of course created metadata associated with it, and they needed to send us this metadata and samples. Then we ran some experiments. In this case, it was genome sequencing, so we generated a lot of data. And we needed to send them back the raw data and metadata, because last but not least, they needed to do the bioinformatic and biostatistic analysis of this data, and then send us back the results. So as you can see here, there is a lot of orange arrows, and these orange arrows for me is a problem. It's an issue because, for example, if one of our partners sent the data into an email, it would have been a big security risk. We could lose some information from one version of a file to another. We could have used some sample ID from one partners and then another sample ID nomenclature for another partner. And so it's a huge mess. It can be a huge mess, have some risk of losing all of the information that you have. Another example that I can show you is another project, not at all the same thing, with an hospital who did a study on vaccines. So they had some samples, they had some data, clinical data, experimental data. Another hospital that did another study, not the same one, not the same number of patients, not the same timeline, everything. Same for the third partner and for the fourth. And so all of these partners sent us their study, each of them, and then we needed to group them together to do a main project, use all of this data together, and try to find something that we wanted to find. For me, still orange arrows, so still an issue, because we still have a risk of security and sharing of data. But here, the main thing was that the data was heterogeneous and that we did not have the same visits, we did not have the same time points between studies, so we needed to harmonize everything. So that's where LabKey came as a solution for us, and I will show you how. So as I said earlier, we wanted to store all of this project data and metadata into one source and one and only. When I speak about data and metadata, I speak about clinical data, sample data, raw data from instruments, and analyzed data. We wanted this secure access, a way to share the data with the partners, and to automatize our workflow, still respecting the FAIR principles. And here you have a quick overview of the second study and how well it went. So we have some assay design, we have some clinical data, we have some data sets and samples, and everything is clean. But how did we found LabKey? So at the beginning, we did some tool benchmarking to try to search how we could harmonize all of this data and centralize it. And LabKey was the winner. We started with the communicative version that we talked earlier about on one project. This project was the one I just showed you with all of the orange arrows. This project went quite well. We did not have any issues with the data, zero discrepancies, and we were happy with our solution. So we said, why not have more functionalities? What inspired us to go from the community version to the starter version were the add-ons such as the specific assays like Luminex or flow cytometry, the quality control reporting, the single sign-on for security, and of course, the extract, transform, and load processing that we found quite easy to work with. At the beginning, when we went to the starter version, we decided to do one project, one platform. It was not the best idea we had because at the end of the day, we had a lot of platforms and we had to maintain them. So it was kind of an issue. So it's been more than a year that we extracted all of our studies, all of our projects, and put them into one centralized LabKey platform. And that's the part where the permission for the users became quite handy. So I just want to highlight the fact that LabKey is really optimal when you have multiple partners. Because when we go back to this example of 4 studies different from 4 different partners, we had the physical samples that came to Bioaster, and then we had also to get the data, experimental data results. So we told our partners, we have one platform and you will put all of your data in it. So they gave us some heterogeneous data, no problem. We use LabKey to curate them, to work with them, and to find a way to harmonize them. Then we had 4 different studies on LabKey that were harmonized, and we use ETL process to just do one. And with the clinical data, we had everything into our hands. So here is the data workflow that we present now at Bioaster. You can see that we have our internal LIMS that is handling the sample data. We extract this information from the LIMS to put them into LabKey. Then we have some scientists that do lab work and they send us their data so we can put them into the LabKey platform. The project manager can have an overview of what's going on for his project directly on LabKey and see how it's going. The bioinformatician and biostatistician started to work with the API of LabKey to directly get their data from it. And last but not least, there is a huge work with the data visualization to work with presentation, papers, reports, and everything. But you see that there is some process going on. So that's where I will do my conclusion and perspective, giving you the future things we want to do with LabKey. So first of all, we'd like to generate some templates so the lab work would be easier to integrate directly into LabKey and to have more harmonization between experiments, different experiments. For the LIMS part, we want to do an automatic integration directly using the API, so we won't have to do data management work on it. And last but not least, we would really like to use RedCap as an ECRF because we are doing it externally right now. And also, we want to help our bioinformatician and biostatistician to integrate a little bit more the API of LabKey directly into their pipelines. If I have something that I think is important to keep from what I just presented to you is that LabKey is a great tool. We have a lot of functionalities that we use, and there is still a lot of them that we don't use, but we want to. We had a really good feedback from our users on the data visualization part because they didn't know how to use script or everything. And they are quite happy with the fact that all of the data is into one source only. And more personally, a lot of new skills that I learned, such as ETL process into LabKey and that kind of thing. And so really thank you, LabKey team, to help us and support us on this journey. So if you have any question, please feel free to ask them, and nice to have done this presentation with you.
Thank you so much, Lucie. Yeah, there's a question has come in here. Wonderful presentation. Do you have to work with your partners to receive the study data in different formats, or are you curating the different formats yourself?
At the beginning, we did the curation ourselves, but it was quite time consuming. So what we did is we created the structure into the LabKey dataset or assays or sample types, and we gave them templates to just fill and put directly into LabKey.
Yeah, here's another question. Any tips for folks just starting out with LabKey study?
Sure. The first thing, and you can also add to this, Steve. The first thing I would say is try to really see the difference between assays, datasets, lists, how you can handle them, and which is the best for the data type that you have.
Yeah, great. I can second that. And as a teaser for my November training, we'll spend a lot of time on sort of how to correctly kind of choose which container for your data. Another question, what's the mix of internal, external, third-party systems that are involved in the data transfer?
I'm not quite sure. Sorry, it's late here, and my English is starting to go out of the window.
Sure.
Do you mean how do we work with third-party softwares?
Yeah, perhaps perhaps the questioner will give a little bit more detail there. But I'm wondering if maybe behind the question is, are you using like LabKey's kind of ODBC features at all? Like, are you using like LabKey internal features, or are you using the API, say, of the external software? You know, what kind of mechanisms are at work here?
Yeah, sure. So I will go back to this slide. So for the LIMS part, we are trying to put into place to use the API from the LIMS into LabKey. For the bioinformaticians and the biostatisticians, they are using the API from LabKey. And for RedCap, we will see when we will be there.
Yeah, right. Great. Great. Thank you so much.